In this video I want to take you through cell organelles again. I'll be discussing the functions of the cell organelles, so that'll be a bit of a recap for you. But importantly, you need to be able to label a diagram or a photograph or electron micrograph of a cell so you can identify the organelles. So I'll be showing you the typical features there. We're going to deal solely with the eukaryotic features. We've already discussed that there are two types of cells, eukaryotics and prokaryotes. The main difference between whether one has a true nucleus or not. So the prokaryotes, even though they have genetic material, they do not have membrane-bound genetic material. In other words, they do not have a nuclear membrane around their genetic material. Therefore, no true nucleus. The eukaryotes do. There are other differences between the two, and we'll deal with that in another lesson. But for the moment, you need to have your handout ready so you can start labeling parts of a cell, and there's two tasks for you to do at the end. Just please bear in mind, of course, that these are typical features of eukaryotic cells. So many of these features you would find in both plants and animal cells, and some are unique to either. Right, let's begin with the very outside part of the cell. With the plasma membrane. Don't forget, you need to call the outside of the cell surface membrane or the plasma membrane, not just membrane on its own. The structure of which we'll discuss in much more detail in another lesson, but for now you should have found out that it consists of a phospholipid bilayer. On the outside of the plasma membrane here, I'm showing that we have a cell wall. In plants, the cell wall is made of cellulose. It's a very strong material. It's a carbohydrate, and that's something that you'll also be looking at the structure of in the topic of biomolecules. Cellulose cell wall. Genetic material is contained with the inside the nucleus. I don't believe I've actually got a label for this one, but this is representing the nucleus. Just take note of the fact that I've tried to represent it with some dotted lines around the outside. That will become clear why when I show you a picture of a nucleus shortly. Somewhere in the nucleus you'll have a dark staining body called a nucleolus. By now, again, you should have found out the function of that. It's mainly to do with ribosome synthesis, ribosomal RNA, ribosome synthesis. These structures here, which I'm trying to show you, represent sometimes people call them the powerhouse of the cell. These are the mitochondria, or mitochondrion for singular. It's a site of aerobic respiration, where we get the production of ATP, adenosine triphosphate. That's something you'll come across several times throughout this AS course, and more so in the A2 course. In some plant eukaryotic cells, we find these structures. I've deliberately coloured them green. Because I'm trying to show you that they contain a green pigment called chlorophyll. So these are the chloroplasts, the site of photosynthesis. These structures here, I could have actually extended it mainly throughout the cell cytoplasm. These pink, purpley, squiggly lines. They're representing endoplasmic reticulum, and the black dots are representing ribosomes. Notice that some of these ribosomes are free floating in the cytoplasm. Some of these ribosomes are attached to the endoplasmic reticulum, and some endoplasmic reticulum doesn't have any ribosomes. We call that endoplasmic reticulum smooth. So smooth endoplasmic reticulum, or SER for short. And the endoplasmic reticulum with ribosomes we call rough endoplasmic reticulum, or RER for short. These structures that just appeared, the little brown ones, are described as membrane or flattened membrane sacs. They're quite small, much smaller than endoplasmic reticulum. These are the Golgi Body or Golgi Apparatus. Their main function, I hope you found, is that they are to sort, modify and package proteins. And they package proteins into very small membraned sacs, which are called vesicles. These spheres, which are dark staining spheres I've shown now, contain a package of hydrolytic enzymes, digestive enzymes. These are the lysosomes. They're responsible for breaking down cell organelles. So these lysosomes refuse with cell organelles and releasing their enzymes. They can also break down cells, and they can even break down tissues. The structure I've just got squiggling away on the left hand side at the moment. The small lines I could have actually put all the way around this cell. These are trying to represent cilia, and the long ones are trying to represent flagella. Now, cilia are responsible for moving objects which the cell might be moving along or wafting along. You might be familiar with ciliated epithelium in the airways of the lungs, which will flick or waft mucus up to the back of the throat, and the flagella are responsible for actually moving single cells as well. Now, you should have by now found out what they're made of. They're made of microtubules, and we'll discuss a little bit more about that in another lesson. And while we're on the topic of microtubules, microtubules are also found as part of the cytoskeleton, which I have not shown in this diagram, the cytoskeleton that extends throughout the cytoplasm of the cell. Microtubules are also found in these two little spinning things, which I refer to at the moment as a centrosome. They are made of centrioles, and they're involved in cell division. Cell division is a chapter that we'll be looking at separately as well. And during cell division, these centrioles migrate to the opposite ends of each cell, where they form spindle fibers, which the chromosomes attach and are pulled apart during cell division. Right. Let's have a look at a few pictures of parts of a cell. What we're looking at here is an electron micrograph of a plant cell, and you can see in the top right hand corner PM is labelling the plasma membrane of two separate cells, and in between that CW represented in the cell wall. So the plasma membrane can even be seen here, not in great detail, but it is visible. Cytoplasm is not labelled, but you should be able to identify that. Dictylostones, I can never say it properly, there's D at the bottom there. That's actually, I think, the plant version of Golgi Apparatus. What else have we got here? Well, what I want you to notice is the letters NE towards the bottom half of the cell. That's labelling a membrane, and actually you can see there's two lines to that membrane, a double membrane. That's representing the nuclear membrane of the nuclear envelope, which surrounds the nucleus. NL in the middle of this picture is inside this dark stained area of the nucleus. That's representing the nucleolus. You may also notice ER at the top, which stands for Endoplasmic Reticulum, although it's quite hard to identify on this particular photograph. PD on the left, plasmodesmata. It looks like there's a line going between two cells, and this is exactly what it is. It's an extension of cytoplasm between two cells, so this is one of the ways that plant cells can communicate. Plasmodesmata. Although we haven't labelled that on our eukaryotic diagram, we will come across plasmodesmata in other topics, such as transporting plants, when we look at active uptake, communication between the phloem sieve tube elements and companion cells. If you ever watch this video again after you've studied the whole year, that will make a lot more sense for you at this particular time. Right, this is a close-up of the nucleus. It looks a bit like a strawberry. What you're looking at there is actually holes in the membrane. It's a double membrane. It's labelled inner and outer membrane there. So it's a double membrane organelle, and it's got these holes or pores, nuclear pores. And that's to allow things in and out of the nucleus. One thing you will become familiar with is the passage out of the nucleus of messenger RNA. That's when DNA is transcribed into messenger RNA, and the messenger RNA moves to the ribosomes for translation into proteins as a result of protein synthesis. Take a note at the scale on the bottom right hand side, 500 nanometers. So this would be an opportunity for you to try and convert that to micrometers. So work out how many micrometers that is. We're looking at a picture here is a mitochondrion. Mitochondrion. Again, you've noticed it's got a double membrane on the outside, and the inner membrane makes these little up and down lines that you can see spreading inwards. So that gives the inner membrane a very large surface area. Those folds of the inner membrane are called cristae, and all the space around those folds within the mitochondrion is something called matrix. We'll look at that a little bit more in another lesson. The fact that you've got light labelled at the top it sort of gives away what organelle this is. This is an electron micrograph of a chloroplast, and if you look closely again, you notice it's also a double membrane on the outside, and the inner membrane forms all those structures that you can see inside. That inner membrane is called a thylakoid membrane, and it starts to fold on itself to make a stack of membranes which are labelled GR there. That stands for granum, or a granule stack. So all those little stacks of membranes is called a granum, and it's made of thylakoid membrane, and it's on that membrane that you'll find the chlorophyll molecules that traps light. The other part of the chlorophyll you need to know here is labelled STR, and that stands for stroma. That's the equivalent of the matrix in the mitochondria. And you've got lots of enzymes there which are going to be involved in photosynthesis as well. CW at the top, I suggest that must be the cell wall. What you're looking at here is a huge extensive extension of the nuclear envelope, which becomes the endoplasmic reticulum stretching into the cytoplasm. So you can see these folded membranes. This is the endoplasmic reticulum, and if you notice the little black bumps on it, they're the ribosomes. So this is rough endoplasmic reticulum. These are also flattened membrane sacs, not as big as the endoplasmic reticulum, so this is one way you can tell the difference between the two. These are quite small. These are your Golgi Body or your Golgi Apparatus, where we get the sorting, modification and packaging of proteins into vesicles. And a final picture I want to show you is these are made of microtubules. This is a picture of cilia. So these are cilia on the ciliated epithelium. Okay, let's have a go at labelling parts of a cell. This is a diagram. You should have this on your handout. I'm going to play this through slowly. The question mark is flashing, so every time you have a go at it and write it down, play the video again, see what the answer is, and then pause the video and try and work out what the next one is. Ribosomes or RER, it's pointing to both, so I think that's quite excusable that you can call one or the other. Pore in nuclear envelope, not just the nuclear envelope, because I am actually pointing to the actual space in between the membrane there. So sometimes such labels can be quite picky. So I'm actually calling this the pore in a nuclear envelope. Again, you could have called this ribosomes or RER, depending on what you called the first one. Nucleus, I hope you got that. For this next one, look at the inner surface area of that inner membrane. Mitochondrion. This next one is folded membrane sacs, quite large. That's the clue there, large, and it spreads throughout the cytoplasm. Smooth endoplasmic reticulum. I may have forgotten what the function of that is earlier. You should have found out anyway for homework. It's synthesis of lipids. This next one, it really ought to be dark in colour. So there's a clue. This one should be dark in colour. It's a membrane-bound organelle. Lysosome, containing hydrolytic enzymes. The fact that this is a dark staining structure inside the nucleus should give it away as a nucleolus. Now look at these objects. There's two of these. They migrate to opposite ends of the cell during cell division. So you're looking at a centrosome there, or in other words, the two centrioles. Well, you should have called this one the membrane, but did you remember to call it the plasma membrane? Now you've got these flattened sacs here, probably a little bit larger in this diagram than they ought to be. So what we've got here are the Golgi apparatus, or the Golgi body. Now this last picture, these are membrane-bound sacs which are made by the Golgi apparatus, which might give you a bit of a clue what they are. These are vesicles. So this is pretending to be a secretory vesicle containing perhaps a protein which has been sorted, modified and packaged in the Golgi apparatus and is now being moved to the membrane for exocytosis, removal of the protein from the cell. Exocytosis is a process we'll look at again in another section. Okay, so that's a diagram. Just assess how you did and what you could do to improve yourself if you did not get them all right. Let's now move on to this electron micrograph now, again with some labels. See how you get on here. This is perhaps quite a tough one to start with. If I can give you a clue, it's containing proteins that may well be moving towards the membrane for release. It's a vesicle. Now for this next one, this vesicle has originated from this particular structure. So the organelle we're looking at there is the Golgi body, or the Golgi apparatus. Clue here, very dark staining organelles. Okay, lysosomes. Okay, this one, well, it's quite a large area down there. It looks like lots of membranes, a bit bumpy. If you was able to run your finger along it, you'd feel lots of little bumps. So we are looking at the rough endoplasmic reticulum. Okay, for this structure now, it looks a bit sausage shape, but have a look closer. It looks like you've got little structures inside it, running up and down. Extension of the inner membrane. Mitochondrion. Okay, these next ones should be quite straightforward for you. Nuclear envelope. Can't see the pores in it this time though. Nucleus. Nucleolus. And plasma membrane. Did you remember to put plasma in front of the membrane this time? You can also put cell surface membrane, but as long as you distinguish between this membrane and the other membranes that you find around organelles of the cell. Okay, assess how you did and think of ways that you could have improved yourself, or ways that you can help to remember such organelles and what they look like in future.